Hi, welcome back to our lecture. Today I'll be starting off with part one of our tour of the cell. And I'm going to be talking about how cells are so important to our life in this world because all organisms that you can think of are made up of cells. The cell is the simplest collection of matter that can live, which means that the simplest living thing that you can see is a cell. Cell structure is correlated to cellular function. There are many types of cells in this world, and we can categorize all of them into two big categories, which are the eukaryotic cells and the prokaryotic cells. And within the eukaryotic cells, there are plant cells and animal cells. They're pretty self-explanatory. Plant cells are in plants, animal cells are in animals. They are very much alike from each other, but there are some differences within plant cells and animal cells, and I'll be differentiating them in our next lecture. Here are some basic features of all cells, no matter if it's a prokaryotic or a eukaryotic. All cells have something called a plasma membrane, which we can see in the picture right here. It's the cell membrane of the cell, and it makes sure that all the organelles are within this cell. And there's also the semifluid substance called the cytosol. It might sound pretty confusing what a semifluid substance is, but in simple terms, it's just everything within the plasma membrane that's not an organelle. And we can see that in this picture, the cytosol is everything here, and it's illustrated in orange. And in this picture, it's everything here that are not the little circle or the nucleoid. And this is a eukaryotic cell, that's a prokaryotic cells. All cells have chromosomes which carry genes and hereditary informations, and that's the reason why that we'll look at our parents because traits were passed down from our parents to us. And all cells have ribosomes which make proteins. And we've talked about proteins 2 lectures ago. Yeah, it is made up of amino acids, and if you want to know more about it, you can look back to our previous lecture. There are many differences between prokaryotic and eukaryotic cells. Prokaryotic cells are only organisms that are bacteria or archaea, and they have no nucleus, which means that their DNA is not in the nucleus because they don't have one. Their DNA is in something called a nucleoid, and it is an unbonded region. And in prokaryotic cells, there are no membrane bonded organelles, or their lack of membrane bonded organelles. And for eukaryotic cells are basically human cells, plant cells, fungi, animals, all sorts of things like that. And humans, we also have eukaryotic cells. Eukaryotic cells have nucleus, which means that our DNA is stored in our nucleus because we do have one, and eukaryotic cells have membrane bonded organelles. This means that all the organelles in our nucleus are bonded by a membrane. And eukaryotic cells are typically bigger or larger than prokaryotic cells because there are more organelles in there. I want to mainly focus on eukaryotic cells and the organelles in there. This is a picture of a plant cell, and that's a picture of an animal cell, and these are all eukaryotic cells. And as we can see, there are a lot of organelles that are in common, and I'll be focusing on these organelles in the following slides. And I'll be talking about organelles that both plant and animal cells do have, and the ones that only plant cells have or the ones that only animal cells have, I'll be talking about that in our next lecture. I'll be starting off with the most important organelle in our human body or in our cell, which is the nucleus. Why is this so important? Because the nucleus contains most of the cell's genes, which means our DNA is in there, all the hereditary information is in there. And if you want to code something, it's got to be in the nucleus. And this is a picture of an animal cell right here. And we can see that this is the nucleus. It is the heart little circle in here, and it is made up of these things, but you probably don't have to know it right now. And all you have to know is that the nucleus is bonded by a nuclear envelope. It's closed by a nuclear envelope. And The second organelle that I want to introduce is ribosome. Ribosomes are particles made up of ribosomal DNA and protein, and in fact, its function is to make proteins. So now you know how are proteins made. They're made by ribosomes. And ribosomes, you can see ribosomes in two locations of a cell, either in the cytosol, it could be like here, 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 anywhere within the cytosol, or it could be bonded on rough endoplasmic reticulum, which we also call the rough ER, and I'll be talking about that in the next slide. Here's a picture of how ribosomes are bonded on rough ERs. The purple thing, the purple little coils and folds are the ER, rough ER, and we see those little circles attached onto the rough ER, and these are ribosomes. And endoplasmic reticulum, which we also call the ER because its name is pretty hard to pronounce, and I'll be abbreviating it as ER. It's surrounding the nuclear envelope, and as we can see in this picture, it's more clear. This is the nucleus, and that's the nuclear envelope right here. I don't think it's illustrated on this photo, but yeah. And the rough ER is not the rough ER. The ER in general is bonded on the nucleus, and it's attached onto it. It's surrounding it. And there are two types of ER: the smooth ER and the rough ER. The smooth ER doesn't have ribosomes on it, but the rough ER does have ribosomes on it. And the ribosomes we're talking about right here, they're the rough ERs because they have ribosomes on it. And rough ER and smooth ER, their functions differ a lot. Rough ER is mainly used for making proteins and producing membranes. We call it the membrane factory of the cell. And the smooth ER is synthesizes lipids, it makes carbohydrates, it stores calcium, and detoxifies poison. And as we can see in this picture, this is more clear. Yeah, this is the nuclear envelope, and this should be the nucleus in here, and it's not illustrated in this photo. And rough ER are the ones without the little dots, which are ribosomes, and the rough ER are the ones with the little dots, which are the ribosomes. Oops, sorry. And once protein is made by the ribosomes on rough ER, they are carried away with little box. You can think of it as a little shipping box, and we call it a transport vesicle. And then we transport these proteins made by ribosomes to other organelles, which then can modify our protein. So you can think of rough ER with ribosomes on it as a factory which produces things, and this thing is protein. And then once it's made, it is packed in a little box, shipping box, which we call the transport vesicle, and it is shipped somewhere else. And where is it shipped to? It's shipped to this place called the Golgi apparatus. It is the shipping and receiving center in a cell, and it receives proteins and other things. And its main function is to modify the proteins produced by ER. And once they modify the protein, it's all good, and they can ship it to other places. You can think of the Golgi apparatus as maybe like USPS, UPS, FedEx, all those shipping companies. That's basically Golgi apparatus. Golgi also manufactures certain macromolecules, and it stores and packages materials into transport vesicles, which are the shipping boxes. And the last organelle that I want to introduce, which all eukaryotic cells have, is the lysosome. It is the digestive department. It breaks down macromolecules, and it is also used for digestion and waste removal. Thank you so much for listening to my lecture today. If you have any questions or concerns, please feel free to email me or comment down below, and I'll get back to you as soon as possible. Thank you.